Welcome to the pre-course lecture for DIY transcriptomics. This lecture is going to be broken up into two separate videos. In the first video, I'll talk about the technology that underlies not just RNA sequencing, but also many different high-throughput sequencing applications in use today. In the second video, we'll discuss key experimental design considerations for when you're planning your own transcriptomics experiments. How deeply should you sequence each sample? How many biological replicates are appropriate? What read lengths should you use? How much will it cost? We're going to cover all of those topics and more. By the time you finish this lecture, you'll understand how the kinds of fluorescent spots that you see sparkling on the screen in front of you, how these are really fundamental to understanding high-throughput sequencing data, and how they're responsible for tremendous amounts of data generation that we'll ultimately spend the rest of the course learning to analyze. Okay, so before we jump into the lecture, let's get a few housekeeping items out of the way in this early lecture. Firstly, whether you're just participating by watching the lectures online on our website, or whether you're also participating in our in-person data labs, you're going to want to watch that welcome video. It really lays out how the course is structured and also gives you a sense for how to navigate the course website. I'd also encourage you to check out our course FAQ page where we post a lot of useful information. For those of you who are participating in the in-person labs, you're going to want to also check out the information page for laboratories. It'll explain what these labs are, how they're different than lectures, and how you participate in these labs. Of course, if you're just watching virtually, you can still look at our labs. After each lab, we're going to post all the information, the questions, and the scripts for those labs, so you'll still be able to get some useful information from looking at those pages. Finally, for those of you who are participating in these in-person labs, you're going to need to check your email for the invite to the course Slack page. As a reminder, all communication for this course happens exclusively through Slack, so if you haven't accepted the invite and aren't on the Slack page yet, pause this video, go find that email, and join the page. You're also going to want to dig up the email from DataCamp that was inviting you to join their site. They're a third-party resource that we use for homework assignments. And again, that's only for those of you who have signed up for the in-person labs. Great, so we're going to jump into the lecture, and this is going to be the sort of format we're going to follow for all lectures. Go to the lecture card, click on the lecture card to open the lecture page. And I just want to highlight the structure of these lecture pages is the same for every lecture. At the top is a link to the slides, actually the actual slides you're viewing now in the video, that you can open in iCloud and follow along, and that gives you access to all the links in the slides as well that I've been talking about. We have a short description of the lecture, the learning objectives, which I'll cover in just a moment, reading material, and this is all just extra material for extended learning. If you're interested in the lecture topic, you're going to find lots of useful information under this reading section. And if you scroll further down, you're going to see the video lectures actually embedded in this lecture page. Okay, so what do we want to accomplish in this lecture? Well, first and foremost, you're going to want to understand Illumina's sequencing by synthesis, or SBS technology. We're also going to learn to set priorities when planning a sequencing experiment. You'll understand the basics of library prep by the time we finish this lecture. Now, this course is not geared towards the wet lab aspects of sequencing, but we'll talk about some of the concepts of library prep, and I'll point you towards a resource that I think will be really helpful anytime you have to carry out experimental or bench aspects of sequencing studies. And then finally, we're going to get familiar with the jargon that really circulates around RNA-seq and high-throughput sequencing studies. After all, jargon can be one impediment to learning and an impediment to starting in a field, and that's no exception for sequencing and bioinformatics. In fact, there's a lot of terms you've probably already heard thrown around: read length, read depth, libraries, paired end, cluster density indices, and more. So we're going to spend a little bit of time in the lecture clearing up this muddy water so that this terminology and jargon doesn't impede your ability to learn. All right, so we're going to take a step back in time and go back to the birthplace of high-throughput sequencing. And like many great ideas and exciting developments in life and in science, The development of high throughput sequencing really had its start at a bar over beers. And in this case, it was the Panton Arms in Cambridge. And the folks having those beers and having those conversations were David Klinnerman and Shankar Balasubramanian. And they're both scientists. They are chemists and molecular biologists who fundamentally were just very interested in the potential to use fluorescently tagged nucleotides to study the process of DNA synthesis and also understand DNA polymerase. And as they continued these conversations, they began to realize that these kinds of approaches using fluorescently tagged nucleotides could not only tell you something about DNA synthesis and DNA polymerase, but could actually give you the sequence of a DNA molecule. And that idea, simple and elegant, really formed the foundation of high throughput sequencing and ultimately sequencing by synthesis that is Illumina sequencing as we know it today. Now, it wasn't necessarily a straight or easy path from these early conversations to what we now know as Illumina sequencing, but it was a path nonetheless, and it's one that's been really remarkable to see. So if we go back just less than 10 years, it was not a field in which Illumina was the clear dominant player. In fact, if you were somebody interested in doing a sequencing experiment, it was actually a bit overwhelming. You had many different technologies. You had Roche's 454 pyrosequencing. You had solid ABI solid Life Technologies sequencing. You had Ion Torrent and also, of course, Illumina. And each of these platforms had its own really technology underlying it, had its own consumables, its own pricing structure, and its own pros and cons, which as a scientist on the user end made it really frustrating to understand which technology was the best and which was appropriate for your question. And ultimately, you often ended up going with the one that your core facility recommended. But this crowded field was not crowded for long. What happened eventually were technologies like Solid ended up like this. This is actually a picture from the laboratory next to the one that I did a postdoc in, where there were two Solid sequencers just simply with tarps thrown over them. Because once a company no longer supports a technology, even the most sophisticated instrument becomes obsolete. The same thing happened with Roche 454, with Roche announcing in 2013 that it was shutting down its pyrosequencing business. And that really left Ion Torrent and Illumina, with Illumina really gobbling up the lion's share of the sequencing market. I like to look back on this article. This is a 2008 Nature article talking about the year of sequencing in 2007, just to really highlight how fast and how far we've come in a matter of a little more than a decade. So in 2008, this article highlighted that since the commercial release of the platform, and in this case that was the Illumina Genome Analyzer, and this technology, by the way, was really first, the technology we just described in the previous slide, was really first developed by Solexa, and then Illumina acquired Solexa. So since the first commercial release of the platform, they have sold 100 instruments. 100 instruments. I mean, that's a number that would be reasonable to have in a university, much less a country or globally. So the Illumina sequencing business, the start of high throughput sequencing, has just developed exponentially. The other important thing to highlight here is that the statement here, the early chemistry work, which again was done by Solexa in the UK, led to the creation of a reversible terminator nucleotide. Right? This is the dye-labeled nucleotide and a modified polymerase that would incorporate it. And that's important because it really, in essence, represents two key pieces of intellectual property that Illumina had at its disposal, a reversible terminator nucleotide and a modified polymerase. And this technology ultimately, and we'll get there in a minute, but ultimately led to some of the first, and this is one but not the only early RNA-seq paper where this sequencing by synthesis was applied, in this case to yeast, to generate a transcriptome. And this is a paper from Michael Snyder's lab at Stanford in 2008. So fast forward to today, we have Illumina dominating the sequencing landscape with a range of sequencers. And the nice thing for you as an end user is that you don't really need to care so much about the differences between these different Illumina sequencers. Really, they all use the same fundamental underlying technology, sequencing by synthesis, which we'll go over in just a minute. But the real way in which they differ is simply on data output, with the NovaSeq generating the largest amount of data output and the MiniSeq on the opposite end of the spectrum. Of course, that also means that they scale by cost from left to right, with the NovaSeq being the most expensive. The advantage of having something like a very expensive NovaSeq is that you're delivering the lowest cost per base. Okay, so let's focus in on a few of these and just talk about briefly how these different instruments are used in laboratories and where they're valuable and where they're not. So I mentioned the MiniSeq and MySeq, both small sequencers that you could easily have, and in fact many people do have in an individual laboratory. A NextSeq, which also has a small footprint and is reasonable to have in a laboratory, and then a NovaSeq, which you might find much more commonly in a core facility. On this end of the spectrum, we have generating on the order of dozens of millions of reads, 1 to 25 million, short reads, upwards of 150 to 250 base pairs, and the whole sequencing run is done in 24 hours. On the other end of the spectrum, the NovaSeq just generates a tremendous amount of data, up to 10 billion reads. Again, only up to about 250 base pairs, but it takes much longer to run. The NextSeq, when it came out, really struck this great balance between low-end and high-end output sequencers, and that it had a range of output that was quite wide and large, generating up to 1.1 billion reads. This is enough to sequence the transcriptome of multiple mammalian samples. In contrast, the MiniSeq and MySeq do not generate enough data to really cover more than one sample for mammalian transcriptomes. And so it's really not adequate for the kinds of transcriptomics we're going to be talking about in this course, but the NextSeq absolutely is. And that amount of data is generated in just 24 hours. I mean, it's really staggering the rate at which these instruments can generate massive amounts of sequence data. So the MiniSeq and the MySeq are really very well suited for a lab, but just aren't sufficient for transcriptomics. They're really well suited for things like amplicon sequencing, and that could be targeted PCR panels, 16S sequencing of the kind you would do for microbiome profiling. It's really good for bacterial genome sequencing and bacterial transcriptome sequencing, and even quality control of very large RNA-seq experiments so that you can check all of your libraries before spending the money on a much larger NovaSeq run. In contrast, the NovaSeq is really best suited for a large core facility where there's a dedicated technician. The setup's more complicated than some of the smaller machines. The consumables are far more expensive. But the upside is there really are endless applications available to you on a machine like the NovaSeq. And again, the NextSeq strikes this nice balance where it can do many of the things that we would expect from a NovaSeq faster, and still maintains a lot of flexibility and small footprint. So the bottom line is, because of this range of options for sequencers, there's been a real democratization of sequencing, meaning sequencing is not just under the ownership of large core facilities that have things like the NovaSeq, but rather small labs, relatively small labs, can have small sequencers and be generating lots and lots of data. As a result of this democratization, there's been a tremendous development of different sequencing-based assays for a variety of different fields of biology. And this is really most sort of almost grotesquely highlighted by Illumina's For All You Seek posters, and I've linked to those down here, where each row in this poster highlights a different application. In this case, the poster for RNA, there's one for DNA, and more recently now, of course, one for single-cell-seq. And if you zoom in on any part of this poster, you're going to see individual sequencing-based applications, ChIRP-seq, CHART-seq, RAP-seq, GROW-seq, some of which were one-off technique developments that really aren't widely adopted, and others that become really foundational to an entire field of biology like epigenetics. So as a result of this democratization, there's been a real shift in really, to be honest, what's required of you as a scientist and what you should be thinking of in terms of where to devote your efforts. And I love this paper, now quite old, from 2011 in Genome Biology, where they really predicted this very accurately, this shift in effort of where time and money are spent on genomics experiments. So, you know, in the early days of sequencing, a lot of money, a lot of time devoted to the sequencing itself. It's very expensive, hard to access, so you didn't plan big experiments with lots of samples. You spent all the time and effort on the sequencing bit, and the analysis was relatively straightforward because, again, what was remarkable was your ability to sequence, not a sophisticated analysis that you'd be carrying out. Fast forward to today, which for this paper was the future, and we're past that future now, but I think it's pretty accurate that now sequencing, because it's so accessible and inexpensive, you really don't devote much cognitive effort, much time, much money to sequencing. And actually the management of the data that comes off those sequencers has been really streamlined, with even the machines themselves handling some aspects of data processing. Instead, you as a scientist now can do huge experiments which require a lot of consideration for experimental design and sample collection. And then on the back end of things, there's a lot of effort that goes into data analysis. And really, this course centers really on this aspect of the work, which is how to handle data that comes from these experiments. We'll talk a little bit about experimental design actually later in this lecture. So the bottom line is, as we've had this democratization and increasing accessibility of sequencers, there's been an explosion in the amount of sequence data deposited to public repositories like NCBI's Sequence Read Archive. And you can even overlay when different sequencers were introduced, and it really nicely highlights how each of these technology introductions really accelerates this process. By the way, I will always, when I show a plot, try to link to the underlying data as well as the code I've used to produce it. So again, if you open up the slides and click on these links, you'll be able to access those. All right, so as I mentioned before, the nice thing about this technology, about Illumina sequencing, is that if you understand the underlying technology of sequencing by synthesis, then you understand how all of these sequencers work. So now let's talk about SBS and what it means. We'll start with the idea of a flow cell, which is, for all intents and purposes, you can think of it as a glass slide. And on that glass slide is arrayed oligos that are linked to the glass. If you have DNA molecules and you prepare them, and we won't really talk about this until later in the lecture, and we're only then going to talk about it very superficially, but if you take DNA oligos and covalently and link them to complementary oligos, then when you add your sample to this flow cell, it will be captured by the oligos bound to the flow cell. Once captured, you can add in nucleotides, polymerase, and you can make a copy of this molecule. You can denature that duplex and actually discard the original molecule that was captured. So that's right. So you're actually throwing away your original experimental sample that you worked so hard to generate. But what you're left with is a copy of that molecule now linked to the surface of this flow cell. At this point, this represented a major technological hurdle for Illumina in the early days. I mean, if you can imagine, one could, in theory, generate sequence data from this single captured molecule. If you had nucleotides that were fluorescently tagged with different fluorophores for each A, T, C, and G, then you could, in theory, get that sequence data as you extended along this template. But that would require tremendous resolution, cameras that could see fluorescent nucleotides moving along this single molecule. And that just wasn't feasible at the time that sequencing technology was first getting off the ground. So how to get around this technological hurdle was a real issue for Illumina. They had their reversible terminating nucleotide, great. They had their modified polymerase that it could incorporate it, awesome. But they needed some way to get around this idea of the fact that single molecule sequencing really wasn't possible at the time. That's where Pascal Mayer's group comes into the picture. He was running a lab at Mantia, which was a company in Switzerland, and they were very interested in solid phase PCR. In this schematic from their original 2003 paper, I think really highlights some of the parallels with what Illumina was doing. They have a molecule they're capturing on a solid substrate using an oligo. The thermometer here represents your PCR cycling temperatures. You cool down that reaction to get annealing. You raise the temperature to get extension along the template, and then you raise it yet again to denature this duplex and get rid of your non-linked molecule. But this is where things get interesting, which is Mayer's group decided that they could take advantage of a second oligo on the substrate if they had another a complementary oligo on the other end of the molecule. And in doing so, they could cause this molecule to bend over to grab a hold of the other complementary oligo and then carry out another extension along this template to make a copy of that molecule in the same location. They could then denature again and stand these molecules up, and now they've got two when they originally only had one. And in theory, they could repeat this cycling, this PCR cycling process, as many times as they wanted in a very controlled fashion, each time doubling the number of molecules, exact copies of the same molecule in the same location on the glass slide. I mean, there's this really great statement from this paper, and I'll just read it here. Solid phase DNA amplification leads to the growth of a colony, a lot like a bacterial colony actually, of molecules attached to the surface and located in the same region. This characteristic could be easily exploited in the design of DNA microarrays. Mayer's group wasn't even thinking about high throughput sequencing. No one really was. So the impact of this kind of discovery hadn't really been realized yet. Illumina acquired this technology. The rumor I've heard is they had to actually rescue the instrument from an auction before it went to the trash heap. I don't know if that's true, but it's an example of a critical breakthrough in Illumina's technology that almost didn't happen. And so that leads to what we now know as sequencing by synthesis, where you capture an oligo. We have a second oligo at the other end of the molecule and its complement also on the flow cell. We then carry out what's called a bridge amplification, and now we have two copies of that molecule, and that process of bridge amplification can be precisely controlled and repeated as many times as you want to generate a cluster. Now, this process is not, there's no data on sequence being collected yet. It's just a PCR amplification to take one molecule and expand it to many so that then imaging becomes a much easier process. Okay, so no fluorescent nucleotides at this point. So in actuality, we get thousands of molecules amplified in parallel in these spots. Each PCR colony or polony, as it used to be called, represents about 5,000 copies of a molecule in a 2 micrometer spot. And what Mayer's early work showed very nicely is that you could precisely control the size of the polony by simply the number of PCR cycles you went through. More PCR cycles, more doubling of those molecules, a larger spot, if you will. And you could control the distance those spots are apart from each other simply by how densely you seeded the solid substrate with your oligos. Okay, and that idea of size and density of spots is really critical to understanding one of our key terms in sequencing, which is clustering density. It's a term you've probably heard before, and it exactly refers to what I just described, how close and how large these spots are. So we can see on this end of the spectrum, each of these white fluorescent spots is a polony. These are very, very crowded, and you can imagine if a camera tries to look down and interpret data coming from each of these spots, it might get really confused and ultimately have to discard much of the data because it can't tell one spot from the next, similar to how if you streaked a lot of bacteria on the plate, you can't pick one colony from the other. So there is a risk of what we call overclustering and losing a lot of data and having a really poor quality sequencing run. On the other end of the spectrum is underclustering and not putting enough material onto the flow cell. And in that case, we get good data. The machine, the cameras can easily distinguish each of these spots and collect good data from those spots, but we just get very little data. So the risk of underclustering is just a decreased amount of data collection. So there is still, by your sequencing center, this sweet spot they try to hit of getting enough clusters that we get good amounts of data, but not so many that we end up over clustering. And that number depends on the sequencer and the flow cells and lots of other things, but it can range from 170 to 220,000 clusters per millimeter squared for the next seq, and upwards of a thousand, so 1 million clusters per millimeter squared for the high seq. Okay, so then what we're talking about then is once we have a cluster, if we focus on one polony here, then once that polony is of the appropriate size, and of course this is all precisely controlled by the sequencing instrument, there is a finite or fixed number of bridge amplifications of PCR cycles that the machine goes through where it knows it will generate polonies of the appropriate size to then collect data from. So once that polony size is reached, then the actual data collection begins. We have a sequencing primer that is added to the reaction. And from that sequencing primer, we get incorporation of the reversible terminator nucleotides that are labeled with fluorescent dyes. We then collect that initial sequencing data. We then get a barcode information and we wash that away as well. Now, if you're like me, you're a visual learner. So what I'm going to do now is play a video from Illumina that is a cartoon animation of how the sequencing happens. I'll stop the video at partway through because we don't need to watch the whole thing, but it will really, I think, hammer home how the sequencing by synthesis works.
Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read.
The index, by the way, is the barcode that's going to tell us which sample each of the reads in our data came from.
After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds this.
Now, if you're doing single end sequencing, effectively your sequencing run is essentially done at this point. In this case, in this video, they're showing you paired end sequencing, where they're going to bend the molecule over and sequence from the other end for a second read.
Second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer.
And that's it. The second read is going to be done exactly in the same way as the first read that you saw earlier in the video. The result from all of this is a text file that contains a lot of data. And so one thing to highlight at this point is that high throughput sequencing data is stored in a special file format called a FASTQ file. Now, many of you may be familiar with a FASTA or FASTA file, and I think it's good to start with a description of FASTQ by first talking about a FASTA file. A FASTA file is really simple. It has a header that starts with this greater than or caret sign, and then has some description of the sequence that it's about to tell you. We also call this a def line. It then just has the sequence, and that's it. And then the next, if there is another sequence entry in that FASTA file, it'll start with another greater than symbol and another def line, more sequence, and on and on and on. FASTA files are how we store things like transcripts. So we'll use a FASTA file as our reference file for the RNA-seq alignments. It's also how you store a genome sequence or contigs. And FASTQ files are really quite similar. They're text files, just like FASTA files. But in addition to a def line or a header, which in the case of FASTQ files starts with an @ symbol, and in addition to having a sequence, just like FASTA files have, FASTQ files are unique from FASTA files in that they also have a quality score. And the quality score is separated from the sequence by this plus sign. So there are 4 lines for every sequence in a FASTQ file. Now, the quality score is encoded in these ASC2 characters. And we're going to talk about why that is and how those relate to the sequence in just a second. So the important thing to highlight, these are just text files. FASTA, FASTQ, they are text files, but they can be really, really large text files, the kind that you would never want to open in a text editor of any kind. Again, 4 lines per sequence. I told you earlier in this lecture that some of these sequences generate, some of these sequencer and sequence instruments generate 1 billion sequences. Well, you would have 4 billion lines in that text file. So why ASC2 characters to encode quality information? Well, the answer is pretty simple. If we tried to encode a quality score as its 2-digit quality number, that would mean that in a very large file with billions of characters, we would be doubling the amount of information from 1 character to 2 anytime we need to talk about quality. So instead, we use this encoding, these ASC2 encoding system to encode a range of quality scores from 0 to 40 using the characters above. Now, most high throughput sequencing, especially sequencing from Illumina, ranges from a quality score, a Q score, in other words, of 30 to 40. So these are the kinds of characters you're going to see in your FASTQ files. Now, a 1 in 30 is a quality score of 30 is a 1 in 1,000 chance that the sequencer called that sequence incorrectly. A 1 in 40 is a 1 in 10,000 chance, and it goes down from there. A Q score of 20 is a 1 in 100, right? So this is an idea of this kind of inherent sequencing error that you're going to find in Illumina short read sequencing. So if we look at that FASTQ file again, we can see there's a stretch of A's here, which are Q score of 32. We get up to F's and ATs here at 31, F 37. And so that's how you can interpret the quality of the base immediately above that Q score. Now, one inherent characteristic of Illumina short read sequencing is that as the sequencing reaction proceeds, the quality of the bases begins to drop off, eventually precipitously drop off. And so we start with almost 100% of our bases being greater than Q30. So, less than a 1 in 1,000 chance that it was called incorrectly. But as we proceed, and this is a cycle number moving from left to right, we can see that now, only 80% of our bases have a Q score greater than 30. You'll notice that at the end of this sequencing reaction, the quality of the reads hops back up again. That's because it's a different sequencing primer, now sequencing that barcode or index. And so essentially we start over with our quality scores. Remember, quality scores are very good when the sequencing reaction first starts, and they decline as the read gets longer. Indexes are really short, so the quality tends to be really, really high. So there are a number of programs that both allow you to evaluate the quality of your short reads and will allow you to trim bases that are low quality or get rid of entire reads that don't have an average quality of some threshold. And we'll play around with some of those programs, and you in fact installed some of them like FastQC using Conda, or will do that in the next lecture. Okay, so that marks the end of this part of the lecture. In the second part, we'll pick up and talk about experimental design.